My name is Dr. Paul Wicks, and I'm a neuropsychologist by training, specializing in genetic forms of motor neuron disease. I was really pleased to see a publication come out in the very prestigious journal Nature Communications about an intriguingly titled research study called the SOD1-mediated ALS phenotype shows a decoupling between age of symptom onset and disease duration. Many years ago, nearly 20 years ago, I spent 3 years of my PhD traipsing up and down the United Kingdom in search of people with SOD1 aligned motor neuron disease as part of my PhD. And it was really challenging to find these people, and many of them had different questions about, what different mutations meant for them in terms of maybe how old they might be, how aggressive their disease might be, and then for other members of their families. And then since then, I've done a lot of work in advocacy and research, and have come into contact with many other families who share the same types of questions. So it was really interesting to see this work. And funnily enough, I actually trained at King's College London. So I reached out to Dr. Sarah Opie-Martin, who joins us today, and she very kindly has agreed to share the results of those research findings and to explain a little bit more in detail about what they mean for research, what they mean for the field, and to sort of reframe some of the very technical scientific detail in this great paper into ways that more of us can understand, digest, and use in our practice in the community. So Sarah, will you please introduce yourself and tell us a little bit more about how you got involved in this interesting research?
Hi, thanks, Paul. So I work at King's College London as well, as you said, and I work on the epidemiology of ALS. So that means that I look for patterns in the disease. So who has it, where they live, the gender ratio, how long people live, where it starts, how it progresses. And so I got involved in this work on the basis of that research that I do. And I also worked for a while on the ALS online database, which is a website that presents curated information from the literature on people with specific gene variants in ALS. And so as part of that data collection, we collected clinical data from the papers and put that in a form that can be, then you can run statistical analysis on it. It's not in text form anymore. And so we had that sort of set of data, and then we asked various different groups for information from their kind of specialist clinical databases, and we queried the Project Mine dataset as well. So Project Mine is a big genetics project, sort of global consortium in ALS, and we put all that data together. We found as many sources as we could for people with SOD1 ALS, because as you say, it's a very rare population, so you really have to kind of go globally to find that data. And so because I was working on those projects, that's how I got involved in this particular piece of work.
Okay, great. So you sort of reconciled data sets from multiple different countries, and I suppose that have been collected over decades, really, and try to sort of put them all into one framework. So what are some of the challenges in that? That sounds like it's quite hard to do, to sort of piece together those different jigsaw pieces.
Yeah, I think the challenges are really things like the clinical definitions have changed over 30 years. So there's a lot more that's understood about, I suppose, what the different subtypes of the disease are. And in terms of, sort of upper motor neuron involvement, which actually I think maybe isn't as much of a problem with SOD1 ALS, because it's a bit better defined, but it is generally something that's changed over time. And also things like family history. So I think one of the questions about this work was about the family history and how we defined it. And actually we didn't use a really strong definition because the data are collected by neurologists. And there is variation between what neurologists think is a family history and isn't. If we sort of took what they said at face value, if someone had a family history, but particularly with a genetic variant like SOD1 that runs in families, I think it's maybe more obvious than cases where people have maybe they have a genetic variant, but not everyone in the family with the genetic variant has the disease or something like that. Whereas I think with SOD1, the families are much better defined. So we took kind of family history as just what the clinicians said it was, or what the paper reported it as it was.
Okay. And it seems like people have different amounts of detail, don't they? So, I'm half Irish, for example, so I've got vast numbers of cousins all over the place. Some people might come from a smaller family. Some people don't know much about their family. And equally, the neurologists sometimes will flip over a piece of paper and draw a whole family tree. And other times I'll just say, do you have any in your family, yes or no? And so presumably you've got unequal amounts of data in all those different cases. So yeah, does that sort of affect interpretation? Should everybody be doing this in a more standardized way in the future, do you think?
I think they certainly should. And I think familial and sporadic ALS, I think is, they're difficult terms anyway, because I think a lot of people don't agree on what they mean or whether you should use them at all. But I think, you know, taking a family in a disease like ALS, where, you know, there is a big genetic component, I think understanding sort of family history or genotyping people, just taking their DNA and just looking at what mutations they have overall, is something that should be done sort of more and more in the future, for sure.
Yeah,
and
yeah. It seems like there's a lot of calls for that, right? That everybody should get to, even if they don't know if they have a family history, because especially now there are gene therapies potentially coming through, even if the chance is low that someone without a family history has something like a SOD1 mutation, if there's a treatment specific to the SOD1 mutation, it's probably better that we know for that small chance of being able to treat it than not testing.
Yes, yeah, I agree. I think the other thing to say about family history, and just to go back to what we collected, is we didn't find it affected any of the sort of analysis that we did. So it didn't change the age of onset, and it didn't change the disease duration. So although it's not a very standardized definition, it didn't actually end up getting used in a lot of the models later on. Yeah. So yeah,
that's a really useful way of doing it, because sometimes when you find people sort of nitpicking, we should have done this and should have done that, actually doing a sort of a sensitivity analysis, a so what analysis, and you go, okay, well, I've tried it every which way and the results are all the same. It's a really useful way of doing things, because you say, well, I've tried taking that into consideration. It doesn't change things as dramatically as one thing. So that's useful. Okay.
Yeah, so I'll start. I've just prepared some slides with some more graphs and things that are easier to show and talk through. So I'll talk through those. So these are my details. This is my email and my Twitter handle if you would want to get in touch about anything after the webinar. And I should sort of disclose and acknowledge that we received funding from Avexis for the work on SOD1. So I'll just start by giving a bit of background. So SOD1, it was the first gene that was discovered that could cause ALS. So it's the first sort of genetic risk factor, in fact, the first risk factor for ALS that was discovered ever in the early 90s. And it's found in about sort of 15 to 30% of people with the familial ALS that we were just talking about, with people with a family history, and about 1 to 2% of people with ALS without kind of a known cause that we sort of call sporadic ALS. There are lots of variants. So the gene itself, I think there are 153 amino acids in the gene. So amino acids are sort of molecules that get put together that make up a protein. And there are sort of over 160 variants reported in the gene, and there are more variants than there are amino acids, because one amino acid could change into multiple different amino acids. And we already know that different variants have different sort of prognoses, and so people with different variants have different disease progressions, and that's already known and has been known for some time. So the graph that you can see on the screen, this is a Kaplan-Meier curve, and it'll come up a lot in the talk, or come up a few more times in the talk. And it's a way of visualizing disease progression, or any time to any event happening. So it could be time to disease starting, or it could be the time between the disease starting and someone dying, or any kind of event like that. And along the bottom, along the x-axis, is time, and along the y-axis is the survival probability, and it's the sort of proportion of people that are still alive at that time point. So in this graph, you can see that the people with sort of A4V, or I call them A5V variants. Confusingly, there are two different ways of referring to them. But with this particular SOD1 variant, if you have that, then your survival is shorter than people who don't have that variant, or they have any other variant that isn't A5V. And so you can tell, because the survival curves, if they're more towards the left, it means it's a shorter time to whatever event you're measuring. And if they're further to the right, it's a longer time. So that's an explanation of the graph, and also sort of background information.
So just a question on that, Sarah. So just to be clear then, so most people that we know have MND don't have a family history, but amongst those that do have a family history, quite a large proportion of them have a mutation, a SOD1 mutation. So sort of a spelling mistake in the recipe book that makes their motor neurons, if you like. But that there are many different spelling mistakes, right? Sometimes there'll be an E for an O, and that's a little bit easier to read than if it's a Z for an O, right? And so we have all these different variants, but they're still variants of SOD1. Is that the right way to read it?
Yeah.
And then depending on, say, how bad the typo is, could mean different prognosis implications. So that might mean they might get it earlier, or it might be more aggressive. It might even be that some people get it in their vulva, their throat and speech regions, more than their arms or their legs. We don't really know fully what these different variants mean, and that's what your project is partially set up to do, right? That's what ALSOD is set up to do, to try and gather all this data together. Does that sound right? Yeah,
that's absolutely exactly.
And on this chart, are we talking about the difference between people who have A4V or A5V, which is the same thing, versus non-A4V or A5V? That's still people with SOD1 mutations, or people with all other ALS?
That is still people with SOD1 mutations.
Okay.
So this was a study done in a SOD1 group.
And in North America, A4V is the most common type of these variants, right? So even though there are 160 variants, they're not all equally distributed. There's a much higher number in the US, for example, of this more rapidly progressing one, which is why we're super interested in these variants in particular.
Yeah, that's absolutely right. And this study was a North American study, and we asked them for their data set that they used in this paper. So the data that we analysed includes this. So another study that was done previously to ours, that looked at the sort of disease progression with different SOD1 variants, was done in Australia. So that was by McCann et al. And this is another Kaplan-Meier curve, which we showed before. So it has the disease duration along the bottom. And this is divided, I think there are 5 variants here. And so you can see the E101G has a much longer survival time than the D125V. So it's already known that different variants are associated with survival time. These studies, they were well designed. They're in a well-defined patient population, and they show things that are really useful and interesting. They're kind of single clinic studies, and so you have the problem that they're not big sample sizes, and it's hard to make kind of good statistical inference. So the first study that I'll go back to, it had a really big sample size. As you say, this variant is really common in North America, but it's only one variant out of the kind of 160. And then this second study looked at more variants, but there weren't as many people per variant. And so this is the kind of difficulty with the studies that existed already. What you alluded to at the beginning is that you need to get a really big sample size together to kind of make these, statistical inferences. And what we also wanted to try and do, which it's debatable as whether we achieved this or not, was to try and work out a way of grouping the variants that isn't by single amino acid change, because it's quite hard to kind of run statistics if they're not grouped into big groupings. So we wanted to get the sample size big enough to work out what the groupings were outside of just this single molecule change, if that makes sense.
So I guess as well with these smaller ones, these might be the same families, right? So this line could be 2 brothers and a cousin, and so they may have other things in common. Maybe they've all been, farm workers or something, and so they're not completely random, and it's just the mutation variant that's causing the change, right? That's one of the risks of relying on these smaller groups. I was going to ask you a question about the naming convention, because people might be interested in this. So if it's E101G, what does that mean? Because we know the letters of DNA are Gattaca, aren't they? So there isn't a typo that an E should be a G on 101 or something. What is it, the E101G and this kind of naming convention? What does What does that mean?
Yeah, so the E101G, so you can see that before that it has a P dot, which stands for protein. So instead of when you look at the sort of genetic notation, it'll often start with a G, the G dot. So it's P dot, and then the E and the G refer to the amino acids. And so there's different ways of writing them. So there's the full name of the amino acid, and then you often have a 3-letter code and then a 1-letter code as kind of a shorthand. And then the 101 is the location of the amino acid on the protein.
So let me see if I understand this right. So we were talking earlier about how misprints in, say, a recipe book might lead to, errors in your cake, but it's probably useful to look at whereabouts in the recipe that is, because, for example, if you substitute, eggs for zucchini, then it's quite difficult to make, cupcakes out of that. Whereas if you're just making a change in the icing or the frosting, maybe it doesn't make such a difference, or maybe if there's slightly less sugar or slightly more baking sugar, it doesn't make that much of a difference. So it seems like these different variants here are both saying where in the recipe is the error, and then what is the error as well, right? So we're seeing that the protein is much more complicated than a simple recipe, right? It's made up of lots and lots of different instructions. And what these different variants are showing us is where exactly in the instructions, what kind of error has been made. Is that how you read it?
Yes, yeah, definitely. I think that's a really good analogy.
Great. Always use cake. Solves everything.
Yeah. So the last thing to say is it's not just the variant that affects the disease progression time. And again, this was something that was known before the study that we did. But it does seem to depend. So gender has an effect, or sex has an effect on survival time. So it seems like men with SOD1 variants, so this is a graph from another sort of clinic population, specialist clinic population, also a data set that we kind of assumed into our larger data set. And this shows that men with SOD1 variants have a shorter progression time than women with SOD1 variants. And I think someone has asked you, Paul, a question about this in terms of our data set. And I've got a couple of slides on that sort of later on, because it looks really interesting, and it's something that we didn't specifically look at. We because we knew this, we did a kind of control for gender in the analyses, so that the survival difference is irrespective of the gender within each variant. But it does look like, if you break it down, it's actually sort of something going on there. So I'll talk about that a bit later.
And Sarah, do we see this male-female split in the sporadic ALS? Like, is this a known thing just across all ALS that males have got a more rapid progression than females, or do you think this is specific to the SOD1? In
a lot of the models of disease progression that I've used of ALS, it doesn't include male or female as a covariate, as a predictive covariate, or as something you can use to predict the survival. So I don't think it really does. I know there are differences with when you group phenotypes. I think it sort of certain onset can be quicker in men or women. So there are interactions, but in general, I don't think it is. So to summarize, what we wanted to do was collect a large kind of clinical data set in people with SOD1 ALS. We wanted to model the survival by variant, and see if we could model it in a way that grouped variants together, not just by the amino acid change that we were talking about. We wanted to annotate the variants, so look at what part of the protein they're in, and other kind of factors like that. And it was really the aim of the project originally was to try and inform clinical trial design. So to look at whether you could potentially use historical controls. So that's when, instead of using an active placebo group or a live placebo group, you use data instead. And also to increase the power. So this just sort of means so when you have a clinical trial, you're looking for a difference between the groups. Your power it's whether you have the right sample size and the kind of you want decreased variability between groups of people, so that you can say that the change that you're seeing is a real change. And that the ways to increase power are to do things like increase the sample size. If you have a larger sample size, then that means you can be more sure that the results you're seeing are, something that's a real effect. Or you can do things like reduce the variability between groups. So say we know that people with A4 or A5V SOD1 have a really short survival. If you put people together in a short survival group and you put people together in a long survival group, you're reducing the variation between people. So your inferences or the conclusions that you can draw from that can be a bit stronger. So we wanted to kind of have that. Those are the aims of the study. So this is these are all the countries that we got the data from. So I think there are about 35 in total. And although there's quite a big sort of spread, a lot of the data comes from Europe and from North America. So there might only be single cases in lots of the other studies. And then just to talk about the data sources again. So most of it comes from specialist clinical databases. And those are the type of studies that have published before. So those three graphs that I showed at the beginning, they're all from specialist clinical databases. The ALS online database was used as a source of data. So some of that, most of it comes from clinical case studies that get published. And some of it in the past was uploaded. It was kind of anonymized patient data that was uploaded previously. And then Project Mine, which is a global genetic consortium, has whole genome sequencing data for about, I think it's about 15 or 20,000 people. And one of the bioinformaticians, so the people that work with genetic data in our lab, extracted that and looked for people with SOD1 variants. And we added that to the data set as well. And so, yeah, so in terms of members of the same family, it's not known for participants. So we didn't really kind of collect that in our data set. We had a really basic minimum data set to try and get as much information as possible. So I think there's a thing a general kind of phenomenon in epidemiology where you can either have very basic data on a lot of people or really, really well-defined data on a small amount of people. And we went for the kind of larger sample size trade-off. And so some of the data sources did tell us, but it wasn't really known for everyone. And the variants that we analysed, although as you said at the beginning, there can be some really big SOD1 families, they tended to be the ones that kind of made it into the final analysis and the main analysis of the paper had quite large sample sizes, which I think reduces the likelihood that they're all from the same family. So they're kind of the strongest inference. And as we said before, the family history doesn't have a specific definition. So that's where the data came from. And then this graph is a box plot. So it's a way of kind of describing the spread of a data set. And each point on this graph is a person. And along the bottom are the variants that we'd spoken about before. And this is ordered by variant. And then on the y-axis is the disease duration. And we've logged this because there's such a big spread of the data. It's easier to visualize it as
and when you say you've logged it, so what does that mean? So you've made a logarithmic transformation. So now the gaps in between these numbers are not all the same, right? So the difference between 1 and 10 is only 10 months in that much of my finger, but from the same space again, it's about 8 years, isn't it? So from the next jump upwards. So there's a lot of information compressed into there, because I guess if you didn't do that, it would just look like a big mess. Is that right?
Yes, yeah, that's basically it. It makes the visualization a lot easier. So it's harder to interpret, but it is easier to put on one graph.
Okay, so just a couple of questions about the box plot then. So there's no significance, is there, between the slight jitter off the midline? So if there's a dot that's just a millimeter or two to the left or the right, that doesn't really mean anything. That's just so that you can still see the dots, right?
Yes.
Okay, and so it's a little bit like a grouping. If you were trying to shoot a target very far away and all the bullets were very spread out, you'd have a lot of variability. Whereas if they're very tight, you might say you have quite an accurate rifle in this case, or you might say there's quite a consistent impact on the duration here. So for example, if we look at this first one that you've got here, there seems to be quite a big spread, right? So there's one individual down the bottom who sadly died after, looks like about 9 months from, I guess, the time of onset or the time of diagnosis?
These were from onset.
From onset. Okay, so that's a really rapid progression. But then there are other people in this first one, the K4E, who had disease duration going up to 8, 9, 10 years up there. So within the box bit is where most of the people are, I suppose. And then at the furthest extremes of each variant are these kind of these cases. So so this is a ton of information compressed into a really small space. Box plots are great, really, for visualizing this kind of data. And the other thing to say about this, which is specific to this box plot rather than more general, is the darker boxes are those variants that have an independent effect on disease progression. So they're the ones that we found to be associated with faster or slower disease course in the analysis. So if they're a white box, does that mean that there's not enough information from this study to say conclusively that this is a fast progressing or a slow progressing variant? So for example, I noticed, let's take the fourth one, the C7F, after the red box that you've got there, because I think we'll go back and talk about A5E in a sec. That looks like it's in around the 10-month realm, but I noticed there are a lot fewer spots for that variant than for others. So does that mean there's just not enough information, or does that mean with the information that you have, we can't say, yes, this looks like it's on the more rapid end of things? I mean, it probably means that we don't have enough information in this study. It might be a power issue that I was sort of talking about earlier, that the numbers are too small, or there's too much variability. It looks like in this case the numbers are probably too small to really tell. So what's going on in these red boxes then? Yeah, so I highlighted the red boxes really just to say that I think something similar to a point that you spoke about earlier about the different variants, and it's about kind of what the change is and where the change is. So in with the A5T and A5V, these are both changes from an alanine residue at the sort of the 5th codon position, and they both are associated with a faster disease progression, kind of independently of other factors that could cause a rapid disease. Whereas over at the sort of 94 codon, which I've also highlighted, there are 3 variants there, but only one of those variants seems to be associated with a faster disease progression. And I mean, it could be so that G94C, that there are fewer people, and there is more variability, but it does seem that that the location tells you sort of an amount, but it doesn't give you the whole picture. Do we know enough about what SOD1 actually is and what it does to really understand a little bit about what these different things are? Like you mentioned, the particular bit of the protein that's been changed, and obviously, I certainly don't have a model in my head of exactly how the thousands and thousands of proteins I'm made of really work. Is there a sense that, well, this bit was the instruction for this bit of the motor neuron, it's the mitochondria of the motor neuron, or it was the myelin sheath that keeps it all working, or if it was some sort of flag for the immune cell to leave this one alone or clear it? Do we really have any sort of translational knowledge on what these things mean, or are we kind of just working backwards and saying, okay, well, we don't know exactly what's going on, but this gives us a clue that we should be looking in these areas more deeply, maybe in animal models or cell models? Mm-hmm. So I think for SOD1 specifically, I think I mean it's an anti sort of oxidant protein, so it clears up the kind of reactive oxidant species in the cell. So I think those are kind of quite toxic, and it has, when it's in the cell, it kind of forms. You have to have 2 copies of the protein, and they form together for it to be functional. And that's the kind of middle of the 2 proteins. And then on the outside, there are these metal binding regions. So they have to bind different chemicals in order to be able to work. And that's really specific rather than kind of overall its place in the cell. But the
V and the A5T, they're found at this boundary where the two proteins join. And then there, I think, I'd have to look at a model, I can't remember the 94s, but there are other variants that we've mapped on that are found sort of nearer the metal binding regions or near other regions of the protein. So it's, yeah, when you're looking at it, I suppose this graph kind of looks at the protein flat. Mm-hmm. It does miss out on that sort of 3D structure that would be important as well.
And these oxidative reactive species you mentioned, these are also called free radicals, right? So these are the sort of trash bags in the cell that, if they're sort of left behind, will, look like the streets of Paris after a strike. Mm-hmm. And so perhaps now, as you explain this to me, I can kind of see that, when SOD1 is reacting well, it's like the bin man and the bin lorry, or trash man and trash truck, and they both need to work in harmony to do an efficient job. But if the trash man, maybe he doesn't have his shoes on because something's gone wrong, he can still get the job done. Or if the brakes aren't great on the truck, they can kind of still get the job done, but maybe the guy's got to run a bit faster and further or whatever. But if both, at that junction where they both need to be working in harmony, if that's both not working, the doors are locked, or the steering wheel doesn't work, or the grab handle for the worker to work onto, now neither of them are doing their job efficiently. And at some point, that process is going to break down altogether, right? And we're going to get toxic accumulation of these trash bags to the point where maybe the immune system's going to go, oh wow, I can't save that cell, I'm going to discard it because it's too challenging to bring back. So as you've explained that now, I guess I can, in my mind, think a little bit maybe about how that's working, although only an analogy, not real trash bags in human cells. But that's helpful. Okay.
Absolutely. So I think the next slide, so this is what I was talking about earlier, the male-female survival patterns that I looked into in a bit more detail on the basis of the question. So I think someone had asked whether the reason that sort of men have a shorter disease progression than women is perhaps because there are more men with kind of faster progressing variants. So it's not anything about the men themselves, it's just that there happen to be more men who have faster progressing variants in our data set. So I've done it, it's quite a sort of quick analysis. It's not, obviously, and this is all completely unpublished and unpeer reviewed and everything like that, but we found, so the variants that we found to be faster, associated with a faster progression, were the A5, TA5V, G42S, G94A, L107V, B149G. And there were 3 that are listed below that were associated with a longer disease progression. So it does go both ways. There are some that are slower progressing and some that are faster. And so I just subsetted our data set based on people with those variants. And I looked at 2 things. So I looked at the gender ratio by subset, because that's sort of more associated with risk. And I looked at the survival times by the different subset, the faster and slower progressing. And it's actually interesting. It does seem to be a bit of both. So there are more men with faster progressing variants. And then when you compare the survival times, that's the graph on the far left, it does look like men and women have almost the same survival time. So that's, what the person was asking, that is kind of, does seem to be the case. But with the slower progressing variants, you go back to what I've called the overall on that table. So our overall data set, the gender ratio is pretty much 1-to-1. You go back to this 1-to-1 ratio and you have women that have a slower progression for those same variants. So it does seem to be both, and it's not really clear why there is this difference. And I think I mean, so on the basis of the question, I think we're going to look into this more, because it could be something to do with the decoupling. It could be some other kind of way that the variants manifest differently. Is, if the disease is different between men and women, and variants might affect that differently. There's it's trying to a long way around to saying we don't know why that is, but it does seem to be a mixture of both things.
Because I know your colleague and my colleague and collaborator and friend, Professor Amar Al-Chalabi, has suggested this 6-step hypothesis, and we don't exactly know what all the steps are, but the idea is that one has a genetic mutation that doesn't give you a 100% chance of expressing ALS in your lifetime, right? Because there's incomplete penetrance. But what it could be is that there might be other steps, and so other epidemiological research looking at things like physical trauma, participation in high exertion sports like soccer, football, as well as certain occupations like being in the armed forces, may be that either there are more males doing it, or that there's some other factors that are associated with both a high risk of ALS, but also maybe even high physical fitness, right? It may turn out that there are some protective useful effects in other areas, and so perhaps it is the type of people who, you know, go into these professions because they're quite fit, that also do carry these other risk factors, unfortunately. But I think, you know, part of what I'm seeing here in this difference may be that, you know, genetics is not the entire story. There's probably other triggers in the story, and so that's why it's important to continue doing sort of environmental exposure effects to try and understand how genetics may be a very important part, might account for a lot of the variance in some people, but it's still not the whole story.
Yeah, that's absolutely true. And there's all sorts of things like we've talked about people who have a genetic mutation. I think you just said just then, people have a genetic mutation, but they don't necessarily get the disease, and that's referred to as penetrance in genetic research. And there might be something about that. But it's possible that there are women who have the faster progressing variants, but they don't make it into this data set because they don't end up with ALS for some reason, and all that kind of thing. And it goes back to, actually, it would be really useful to genotype a lot more people, whether they have these diseases or not, and just understand how many people are actually, because you know, our data set's biased in the sense that the people already have the disease, and then you do the genetic testing. So yeah, it would more research into this kind of thing, and looking at other factors would be really, really helpful to understand it. So I just to mention briefly as well the kind of further work that we did using the data set, because it was sort of a big kind of data collection project, and one of them was a study of the kind of protein it was a protein physiological study by a colleague of mine at King's. And this is kind of what we were talking about with the looking at where the variant is in the protein, and sort of what the change is. So this interaction between location and change. And she did a modeling study. So it's a simulation study of variants, of different SOD1 variants that we have in our data set. And it was looking at things like how flexible they are, and what shape they are when they fold up with the variant. And she kind of characterized them into wild type like variants. So that means the kind of benign variant that so the wild type is what you would describe the SOD1. It's still a variant of SOD1, but it's one that doesn't cause disease. And so it's kind of benign in that respect. And they're the metal binding region variants, so variants that have a big effect on the metal binding region that I was talking about earlier, and looked at the sort of the onset and the survival between those two different groups. And the disease onset wasn't really different between the two of them, but the survival was, and it was kind of the way around you wouldn't expect. So the wild type like variants, so the ones that are most similar to the protein that doesn't cause disease, had the shortest survival, although that does include, you know, A5V. And the ones that had the change to the metal binding region actually didn't have as much effect at all on survival, which is quite surprising. So I mean, it's only measuring certain characteristics, so it could be that it's not measuring something that has a really big effect, and we're just missing that. But it's also that you wouldn't know unless I don't know, common sense would make you think that variants that have the most change would have the most effect on the disease progression, but actually it's kind of the other way around. So it was quite an interesting study in that respect. Why it does this is we're not sort of completely sure, but.
And presumably there are, you know, our body is a whole sea of compensatory mechanisms, isn't it? And so it may be that the more subtle error skips through some of the error detection processes that exist within the body, whereas the more obvious issues, you know, have a more you know, attenuated response of some kind, whether that's autoimmune or, you know, bits and pieces self-destructing when they know the mechanism is not working correctly. or we often see other systems come into play, don't we? We say, well, if you know, this system isn't working particularly well, we'll compensate in some other way. We see that in MND in the brains of people, different brain regions activating that don't normally activate, sort of in compensatory responses. Okay, great.
Yeah, I think so. Yeah, you could be right. It could be a really misfolded protein would just get shuttled away by the sort of clear up, you know, functions of the cell. And then the other thing to just mention was someone in our team made a kind of a web browser for this data set, so that you can say if you have variants that you're interested in looking at in more detail, or kind of controlling for different factors, or you had your own data set that you wanted to upload, you kind of add it to that. So this work has led to those projects. So just to say that's the end of, those are the end of my SOD1 slides.
Well, thank you. So, Dr. Sarah Opie-Martin, it's been a pleasure hearing about your work, and I think you've answered a lot of questions and certainly furthered my knowledge about how this works. So we're going to share your email address and your Twitter handle on screen here, and then if other people are interested in follow-up questions, they can get in touch. But you know, we would sort of certainly recommend, you can see here, if your research participation in a study or other members of your family has been something you've been committed to in the past, we really want to show this is the type of benefit that comes across, right? This is the sort of deep understanding. We know sometimes if you're just giving your data or giving your blood, it can all seem like it's sort of going out into the ether, but you know, there are very smart and dedicated people here like Dr. Martin, who are constantly working, trying to get to the bottom of this really difficult mystery, and hopefully with our continued efforts, we will untangle this thing, get to the bottom of it and defeat it. Thank you very much.